1 year ago, a baby was born with a disease so rare, doctors had never treated it in a human before. His doctors built him a drug from scratch in about 6 months. Here's how he's doing now, and how far the science has come since. Quick recap. KJ was born with a condition called CPS1 deficiency. Ammonia builds up in the blood, and it's fatal if untreated. There was no existing drug that fit his exact mutation. So a team at Children's Hospital of Philadelphia designed one specifically for him and delivered it as 3 infusions between February and April 2025. Here's the key difference from classic CRISPR. CRISPR works like scissors. It cuts both strands of DNA, and the cell repairs the cut imprecisely. KJ's drug used base editing instead, which works more like a pencil eraser. It chemically converts one wrong letter out of 3 billion in place. No cut, no gap, no repair needed. This is Children's Hospital of Philadelphia's own account, published February 24, 2026, their official 1-year update on how KJ is actually doing. The update itself: he handles more dietary protein than before treatment, needs less nitrogen scavenging medication, and has better ammonia control, even during childhood illness. He's hit developmental milestones, walking and talking. No serious side effects reported. But his own doctor's framing matters here. This is not considered a cure. KJ remains under close, long-term monitoring. And the same tool is already scaling up. Beam Therapeutics is using this exact base editing approach on sickle cell disease. Their Ristocell program finished dosing in its phase 1/2 trial, with data published in the New England Journal of Medicine this April, and a filing for approval possible by year-end. They're also running it against alpha-1 antitrypsin deficiency, where they've already dosed the first patient in a global pivotal trial. There's a third tool, even gentler than base editing: epigenetic-editing. It doesn't touch the DNA sequence at all. It just switches a gene off, like laying tape over one paragraph, reversible in principle. Tune Therapeutics just used it to silence hepatitis B inside liver cells. The first clinical evidence that epigenetic silencing actually works in a person, not just in a dish. And the regulatory side moved too. On February 23, 2026, the FDA published what it calls the Plausible Mechanism Framework, the first formal, repeatable approval pathway for therapies built for one patient at a time. Before this, a drug like KJ's had no clear path to approval at all. Now there's one. Find the exact genetic defect, show the drug actually hits it, and compare the outcome to how the disease naturally progresses. No giant randomized trial required. The honest caveats: this is still early, 1 patient, 1 year, with long-term data still ahead. It's not a cure. Building a drug for exactly 1 person is enormously expensive. It only works when there's one clean, druggable genetic typo to fix. And most rare diseases still don't have one. Thousands of conditions have no known fixable target at all. So what does this actually mean? The case for this proves bespoke medicine is technically possible, and getting faster, not slower. A real FDA pathway now exists for this whole class of therapy. And the same tools are already scaling to bigger diseases: sickle cell, hepatitis B, alpha-1. The case against the economics of building one drug for one patient are still unsolved. Every case still takes months of bespoke work, and most rare diseases still don't have a target this clean. A year ago, this was one baby. The real question is whether the system around it can scale as fast as the science just did. So, a year ago, this was one baby. What's the next disease you'd want this aimed at? Comment below.